Hi, my name is Ty Young, and I'm a member of the University of Wisconsin-Madison's Botany Department, where I study in the Maeda laboratory. In the Maeda lab, we like looking at plant chemicals, and today we're going to show you a very specific type of plant chemical called pigments. So pigments are what give plants all of their unique and exciting colors. It's the green you see in these leaves, it's the red you see in this pepper, and it's the orange you see in this carrot. And yes, as a plant scientist, I do have a lab coat filled with all plants and fruits. But without further ado, let's get started on some of the exciting activities you'll get to do with your own made-at-home plant pigments. So before we get started, I'd like to say just what we're going to need today to isolate our pigments. A strainer, our coffee filters, plenty of containers for catching the pigments, a knife for slicing up the fruits, some filtered water, and some wipes and paper towels to clean up afterwards. You'll also see I have a cutting board. The fruits you'll need are the blackberries, a red pepper, a bell pepper, or any other red pepper will be fine, and some beets, either pre-cooked or raw from the grocery store. With all that stuff, you'll be ready to go. Alright, so the first pigment we're going to isolate today are Betalins. This is what gives beets their deep red color. And as you can see here, I have a beet that I've sliced open. You can see, of course, it is a nice blood red. So the first thing you're going to want to do for isolating the pigment is you just want to chop it up. I mean, I'm using pre-cooked beets, so really I could be using a butter knife right now. If you're using a sharp knife, make sure to get some help from some parents so that you don't accidentally cut your fingers. There's no particular science to this, so don't worry about getting it all into even-sized chunks. You want to take one of your containers that has some filtered water, and you want to add your beet slices to this. And you can mash this with a fork. I like to expedite the process or make it a little bit faster by just blending it for about 3 seconds once we added some of those beet chunks to the water. Sorry for the sound. Nice. Make sure that's nice and mixed up. Good. Then you're going to take a container that you've placed your coffee filter in, and you're going to pour that mixture in there, making sure it all stays in the coffee filter. After that, you can pick up your beets and just gently squeeze so as to not break the coffee filter, because they are pretty flimsy, and you'll have a nice amount of Betalins that are isolated from that. If you'd like some more, just repeat the process as many times as you want to get as much pigment as you might need. So the next fruit up we have are blackberries. Blackberries make the pigment anthocyanins, which give it this nice purpley red color. Now, anthocyanins are likely used to indicate to herbivores or plant eaters that this fruit is ready to be eaten so that the seeds can be dispersed elsewhere. So getting anthocyanins out is really easy. The first thing you want to do is you'll take them and put them in a container, put them in the microwave for about 30 seconds, and then you want to mash them with a fork. Luckily, I've already done our microwaving here, so I'm just going to show you a little bit of mashing. As you can see, a lot of pigment will come out and get suspended, just hanging out in our dish here. And then, of course, we're going to have a pretty familiar technique that you might be aware of, which is coffee filter and container. You'll take your slurry, pour it in. It'll probably smell pretty good because these are microwave blackberries. And then you're just going to do like we did with the beets, take it and squeeze, trying to make sure your coffee filter doesn't break. And you should get a really good amount of pigment. As you can see, it'll be nice red and dark. And yeah, that's easy as 1, 2, 3 blackberry pigment isolation for those anthocyanins. Okay, so last but not least, we are going to be looking at isolating the pigments from red peppers. So red peppers actually get their pigment from carotenoids. Carotenoids are the pigments responsible for the oranges, yellows, and reds that you see in fall on the leaves on the tree. So they're actually always there. It's just that they're only present after the tree stops making chlorophyll, or the green pigment that you regularly see in the tree at other times of the year. So carotenoids in pepper are like the blackberry. They're trying to signal that, hey, Now is the time for birds to come eat and give away these seeds. So here is what the seeds of the inside of the red pepper look like. You usually don't want to be eating those, but the pepper turns red or yellow or orange, depending upon the type of pepper, to let birds know, hey, it is time to come eat the pepper. They eat the pepper and they can carry the seeds long distance while the birds can't digest them, just to make sure that then those seeds are dispersed over a wider area. So moving straight into the pigment isolation, you'll see I've already done a lot of slicing and dicing to take care of this pepper. The first thing you want to do is pick up a good amount of it. Doesn't have to be all of it, of course. And you're going to want to add, I like to use just a little bit over 1/4 of a cup of rubbing alcohol. So doing that very quickly. Another pour. And then you are going to want to just mix them with a fork over the course of about 2 or 3 minutes. All right, so after a good bit of shaking, you should get a good amount of suspension in the rubbing alcohol from the peppers. If you need a little bit more, it's not coming off as easily, feel free to microwave it in a microwave-safe dish, the rubbing alcohol and the peppers, for around 20 seconds, just to get it boiling. That'll help some of the pigment release a little bit better. But now you want to get away from the rest of this paper to just have this pigment. You are going to slowly, through the strainer, pour the rubbing alcohol so as to catch some of the larger particulates. And it doesn't have to be perfect. As you can see, I'm doing it over the cutting board because I know I am the type of person that makes a mess. So feel free to get that out of there. Dab it off just a little bit. And you should have your pigment isolated. Some people will get a little bit redder. Some people will get maybe where I am right now, more of an orange color. I also did this with other colors of peppers, not just red, and I was able to get a really nice yellow off of some of those. So you can see the red gives a much more orange, closer to red, whereas the yellows can really give a nice lighter color. So that's all you'll need for isolating today's pigments.
And I am a postdoctoral research associate in the Maeda lab. And now that you guys have made your pigments, we're going to test these different pigments to see what sort of different properties they have. We're going to start out simply with an acid-base test. So acids are things that are typically sour, like lemons or limes, and they're have certain properties. So when you put an acid into neutral water, it produces hydrogen ions. So these are positive ions that can interact with other chemicals. In contrast, bases, things like soap, tend to be bitter. And when you put bases in water, they produce negative hydroxide ions. And so these hydroxide ions can kind of counterbalance positive ions that acids produce. So now we're going to do the test for the pigments. Actually, regular vinegar is pretty acidic, so we're going to use a little bit of that vinegar for our acid solution. And then for our basic solution, we're going to use regular baking soda. So 1 teaspoon baking soda, and we're going to fill it up to 100 mils. Now, if you don't have a millimeter measuring cup, it's probably about 1/3 cup, somewhere between 1/4 and 1/2 cup. So I'm going to use our distilled water. So I actually have some here, some baking soda solution that I already made, and vinegar here. I have them labeled on the side so I don't get them mixed up because they look pretty similar. And now we're going to do the test on our pigment. So I'm going to take each of the pigments and pour them into these clean jars. We're going to see if any of these pigments react. Now, the reason that pigments have these nice colors is that they reflect and absorb different wavelengths of light. So, for example, your Betalin or red beet pigment absorbs certain colors of light, but then reflects this nice purpley red color. I want you to think about if the pigment reacts to an acid or a base and the chemistry of that pigment changes, what do you think might happen to the pigment? Okay, I'm going to start with the baking soda or the basic solution, I'm just taking a regular teaspoon and I'm going to add 1 teaspoon to each pigment. You might have noticed that the carotenoid didn't change much, but the anthocyanin got a little bit darker. Okay, it's more purpley now than it was. And then the betalin pigment also stayed the same. So which pigment reacted to the acid-base change? Well, it appears that the anthocyanin pigment did because it changed color. So that reaction with the base somehow changed the conformation of the pigment so that it reflects a slightly darker color than the original pigment, which you can see here. So, but what if we add an acid back to sort of neutralize that base that we just added? What do you guys think will happen then? So I'm going to add a teaspoon of vinegar. It's going to bubble because your acid is reacting with the base. So it's kind of neutralizing those negative ions that I talked about. Change. So again, carotenoids didn't change much. They still have that orangey appearance. And the betalins from the beet also didn't change much. They look about the same color as they did. But now the anthocyanin is starting to look a little lighter because the vinegar is actually counteracting that base. And if we add even a little more vinegar, then we might get it to change back to about the original color that it was. So that's it for the acid-base test. And now you're going to learn more about different properties, polar and nonpolar properties of these pigments to see how they can further be distinguished.
Hi everyone, my name is Bailey Clavin and I am also a member of the Maeda lab. Another chemical property we can test at home is the pigment's polarities. For the most part, pigments can be defined as either being hydrophilic or hydrophobic. Hydrophilic means water-loving, and if a pigment is considered hydrophilic, it is a polar substance which is attracted to water. A good example of a polar or hydrophilic substance is table salt. This is because it is easily dissolved in water. Now, unlike hydrophilic characteristics, hydrophobic means water repelling. And if a pigment is considered hydrophobic, it is a nonpolar substance which is not attracted to water or cannot be easily dissolved in water. A classic example of a nonpolar or hydrophobic substance is vegetable oil. This is because when the oil is added to water, it never really fully mixes. Now that we have a better understanding of polarities and hydrophobicity, we can actually figure out the polar properties of the pigments that we extracted from earlier using a simple oil-water method. Now for this experiment, you're just going to need your 3 pigments and a taller container that is filled halfway with either water and then the other half with vegetable oil. Essentially, all we're going to do is pour the pigment into the solution and see where it goes. If a pigment is polar or hydrophilic, it will dissolve in the water section. And if a pigment is nonpolar or hydrophobic, it's going to stay within the oil section of the container. Awesome. So let's start out with the betalins. I'm just going to use this so it's easier to see the differences. So now, once we test the betalins, we're just going to watch to see where the pigment goes. After watching where the betalins went, do you think that they are hydrophilic and polar or hydrophobic and nonpolar? Feel free to pause the video if you need more time to think. Now, since the betalins went immediately to the water section, this means that they are hydrophilic and polar since they were attracted to the water. Next, let's try the carotenoids and the anthocyanins. the colors have settled, we can tell that the carotenoids stayed within the oil section and that the anthocyanins traveled straight to the water. This means that the carotenoids would be a non-polar compound, while the anthocyanins would be a polar compound. Awesome, so now that we know that anthocyanins and betalins are both polar and carotenoids are non-polar. Sweet, so now that we've decided or figured out what all of our pigments are, we can actually use the pigments again to paint with. So once you're done testing all of your pigments through our experiments, feel free to create some fun art with the pigments that you extracted from. And thanks for joining us, and we hope you enjoyed discovering differences in plant pigments.